Inside of our bodies, we have this thing called an extracellular matrix. It's like a little felt blanket underneath all of our epithelium. And cells have to stick to that as well. And it makes sense that we have a protein that's involved in sticking to that particular structure. But interestingly, why do quantiflagellates have that? They're single cell, right? They don't have this external felt work of proteins to stick to. What we think is going on there is that quantiflagellates evolved integrins to stick to the bacteria that they were filtering out of the water. So it's part of a feeding apparatus. And now it's been co-opted and reused to make us more sticky. Appropriately sticky inside. Okay, so here's this cool stuff. You can see where it came from. It came from protists. And it's part of us. So that's what I mean by this background radiation of the Cambrian explosion. We can see traces of these key innovations that arose in distantly related creatures. Don't actually use it for multicellularity. Here's another one. This is another cool animal. I'd like to say it's one of my favorite animals, but when I think about it, every animal is my favorite animal. So Trichoplax is a very cool creature. It's a tiny little 1 millimeter around, roughly, flat blob that lives in brackish or marine water. And what it does is it just crawls around and digests bacteria, algae, whatever it can find in its environment. It's a spectacularly simple animal. This is a cross-section through it. When they've taken these apart, what you discover is they only have 4 cell types. That's it. We have hundreds. They have 4. Some of them are involved in an epithelial surface, so they're just a flat sheet that sits on top of the animal. There's an elastic sort of cell that's involved in contractions, since we get muscles of the animal. And then there's a couple cell types at the bottom, some of which secrete digestive enzymes. So it just crawls around, squirting out digestive enzymes and reabsorbing the digestive material back. It's a great lifestyle. Okay, we know about the relationships. These are very, very primitive animals. So this is like looking at the first multicellular animals that emerged from a Cambrian explosion. There's not much to them. It's just a couple of things working together. Well, when you take apart their genome, though, you discover some surprises. So here's some big surprises there. Can you find interesting proteins? And yes, they're very cryptic, I know. If you take developmental biology from come to University of Minnesota once, I teach a course in developmental biology. You're all welcome to come. It's all about these. Those cryptic notations up there are a set of molecules that are involved in cell signaling. Just like that receptor tyrosine kinase, only we've got a whole bunch of new ones that are involved in cell signaling to one another. So when I see those, it's kind of weird for me because I know about gastrulation in us, and we use those same molecules. So this is stuff that's going on inside. These are key molecules in your development. The other surprise is a full suite of ion channels. Now, ion channels are what we use to generate currents across cell membranes, which, as you might guess, is really important in your brain. What your brain has is cells with these little channels that let through potassium or sodium or chloride or calcium, whatever. And again, I won't give you a detailed description of neurophysiology. We need those to make our brains work. Trichoplax has them. Trichoplax does not have a brain, all right? So it's obviously using them for something else. We think what it's using them for is for salt balance within the cells. And finally, what we find then is a complete set of proteins involved in synapses. Now again, you think about your brain, you've got all these cables going all over the place, all these axons and nerves running all over the place, and they terminate on other cells. And when they terminate on other cells, what they make is what's called a synapse, which is a specialized connection that will then spew out transmitters, chemicals that trigger the other cell to do something interesting. Much of modern neuropharmacology, all those cool drugs that pharmaceutical companies are trying to sell you to make you happy and cure your depression, are involved in manipulating this structure, the synapse, and the molecules coming out. Again, it's central to your brain. It's in Trichoplax. how weird is that? So here are these little creatures crawling around. They've got these precursors to what we will use in our brain. But they don't have a brain. They're not using them for a brain. What they are doing is they're using these for other functions. And we think that what's going on here is this is part of the machinery that the animal uses to secrete digestive enzymes. So more work needs to be done to figure out exactly what it's doing. But we think what they're doing is they're secreting digestive enzymes, and they're using the machinery we use for a brain, which sort of implies that our brains are overgrown stomachs. That's what's going on in our heads. We've shackled this machinery and reused it in novel ways to send signals between cells inside the brain, and instead of digesting, we hope it's not digesting. Okay, so what we discover is when we look at these different organisms, we find that evolution is recycling lots of stuff and combining it in new ways and generating new capabilities. That we have, for instance, machinery in triple plaques that's used for triple plaques-like things, but then can get co-opted and used in complex ways to make things like a brain. So what you should be thinking right now is this question. Does this mean that evolution can't make anything new? That all we do is recycle the old stuff? If that were true, that would sort of suggest, well, maybe the creationists have something there. If there's no mechanism that can create novel proteins, novel structures, then I would be a creationist. So obviously, I'm about to tell you that there are ways. I'm going to show you a very complicated diagram. Don't be intimidated. This is a couple of genes in the ice fish. And if you want to know more about this, I really recommend. Okay. Am I better? Am I louder? Okay. Oh, now there's an echo. All right. So if you want to know more about this, I recommend Sean Carroll's book, Making of the Fittest, which talks about these animals in great detail. The ice fish are some cool Antarctic animals. They're little fish that live in very, very cold water. The Antarctic Ocean is salty and cold, okay? So what that means, if it's salty, is that it lowers the freezing point. So you can have water in the Antarctic that's at -4 degrees centigrade. You've also got fish swimming in this. As it turns out, fish are less salty than the ocean. They're closer to fresh water than salt water, which means their freezing point isn't depressed as much. Which means that if you're an ice fish and you encounter a little patch of -4 degrees centigrade water, you ought to freeze solid like a little ice cube to drop to the bottom and die, right? You ought to instantly freeze. They don't. And the reason they don't is that they are loaded with antifreeze proteins. Just like you put in the radiator of your car. These are small molecules that inhibit the nucleation of ice crystals and prevent ice from forming so that the fish can stay nice and flexible and fluid. Well, the antifreeze protein, it turns out, is fairly simple. Up there at the top is a diagram of the antifreeze glycoprotein gene. See the blue stuff there? What you find there is a whole bunch of repeated sequences. Each of those is for an antifreeze protein. The antifreeze protein itself is just 3 amino acids. It's threonine, alanine, alanine. That's all it is. Simple, small, little molecule. So there's a bunch of copies there in blue at the top there. And also there's a little sequence at the beginning, which is a regulatory sequence that says, okay, here's how you switch these genes on. Here's how you turn them on so these are expressed. Well, they sequence that. That's cool enough. Here's the antifreeze protein. Where did it come from? Well, when they looked at the genome, they were able to figure out where it came from. And that's the second diagram. What that is another gene, a different gene in this fish. It's a trypsinogen-like protease gene. This is a gene that is switched on in the pancreas of the fish. And what it does is it produces this enzyme that helps digest proteins. You've got similar enzymes in you too. They're also produced in the same way by the pancreas. And this is what happens when you eat. The food goes in, the pancreas produces these enzymes, squirts them onto the food, and it helps break them down. And the fish have those too. There it is.